I'll pass over to Jeff, who will give us a bit of an overview of the Australian Biocommons and what they do and how they can help you.
Excellent. Thanks, Kelly. You can see my slides. Excellent. Well, very nice to meet everyone, meet you all. I'm going to, I guess, give a fairly high-level overview of what the Australian Biocommons is and some bioinformatics infrastructure we operate that will be hopefully of use to support the framework initiative, the fungal framework initiative. So who we are. So as you know, NCRIS funding has gone to Bioplatforms to build infrastructure in the omics space. And some of that funding has gone towards the establishment of the Australian Biocommons, which is the project I'm from. And our job is to really provide freely available bioinformatics infrastructure for Australian researchers. And the infrastructure is designed specifically to support collaborative omics-focused life science research. So framework initiatives are an excellent illustration of that. So I thought before we begin, I would just like to get a little bit of an idea of how you all classify your bioinformatics skills or the skills of people in your team. So um, would you classify yourselves as occasional casual users of simple bioinformatics services? So that might be something like you might do a blast search at NCBI or something like that. Or maybe you are complete experts and you're coding and scripting and developing pipelines on a day-to-day basis for your bioinformatics tasks. So if people could just maybe put the numbers 1 to 5 on how you self-classify into the chat, that would be a great way to start off, and we'll just, I guess, have a bit of an idea of how people classify themselves. Got a couple of 2s and 1s and a 3, 4, 5, 4. Excellent. So I'm seeing a bit of a mix, probably mostly around 2s. And some elsewhere. So thank you. That's very useful. So I guess what we're going to talk about is systems that should be able to be useful for everyone, no matter what your bioinformatics skills are. So just quickly about how the Biocommons operates. So we have been in existence since 2019, and we've done a lot of engagement across life science researchers to find out what their blockers have been in bioinformatics infrastructure and what we may do, what we may be able to deploy to help them. So we have engaged through different methods. So we focus on broad groups that are interested in a technique such as genome assembly or annotation, so on and so forth. We engage with research consortia, and I hope this is the beginning of a long and productive relationship. So a lot of these research consortia, as you'll see, are affiliated with Bioplatforms Australia, but we also engage other research consortia that are national and are independent of Bioplatforms Australia. And so there's a couple of examples here we're engaging within the human genomics space. We also engage with the data production facilities. And again, a lot of these are probably known to you and are supported by Bioplatforms Australia, but we also engage with facilities outside of the Bioplatforms network. So it's really our job to hear what the challenges are from the research community and then work with a whole bunch of computational providers to build a more fit for purpose data analysis ecosystem. And the groups we in that, the computational providers, some of these are going to be known to you. So we work with pretty much all the main computational providers in Australia for research. So Pawsey and NCI run the 2 high-performance computers in this country. AARNet run the network. AAF run the Access Federation. ARDC run the Nectar Research Cloud. We also work a lot with commercial providers as well, because that's definitely part of the mix in how people undertake bioinformatics these days, and QCIF and the University of Melbourne. I'm not going to talk too much about it today, but we also collaborate quite closely with groups in Europe. And so, for instance, we're working actively with the EBI at the moment to get their instructions and make it easier for people to submit their data to those repositories at the end of their research project. So we work with all of these groups and we establish services, and those services are some examples here, and we'll talk a little bit more about some of those today. There's quite a few, but they all, I guess, fulfill or fill a gap that we've identified in our consultations. And again, we operate these services in collaboration with a whole bunch of institutions around the country. It's important to note that all of these services are fully subsidised through NCRIS for any Australian researchers. So these are all there, they're well resourced, and they're just available ready now for you to use. We'll go through some of these. So I'm going to talk around or classify them, I guess, by a few different types. So the first are services that are around data discovery and management. And we'll talk quite a bit on data services for data analysis. And I'm going to touch on bioinformatics training and also some community activities that we coordinate. So with regards to data discovery and management, we'll start with discovery. And the first service that we operate, and we do this jointly with the Atlas of Living Australia, is called the Australian Reference Genome Atlas. And what it does is let you find out what's out there already, what data for Australian fungi already exists. So this is just showing you can browse by taxonomic group, and I'm clicking here on fungi. You can see here that 4% of Australian relevant species have a genome already, and 22% have some kind of genetic data. So that might be a barcode, for instance. And this is an interactive viewer, and you can click on this and explore. So I think I'm going down the phylogenetic tree. This is Puccinales, I guess that's how you say it. And I think once we go on one of these, we can get to, well, for this one, there's a full genome. Click on view, and it's possible then to see all the stats on that genome assembly, but also I think I'm going to say, yes, you can get it. So you can just click on that and that goes straight from the source. In that case, it was NCBI, but we're also building this system so it will be able to interact or pull data directly into some of the data services I'll talk about brief in a minute. The next is something you're probably already aware of, and that's the Bioplatforms Australia Data Portal. So this is available for your framework. I see Mark Turles on the call. He is one of the people behind operating this, and it's for managing the omics data that's produced by your framework initiative. So when I say managing, I mean storing the raw data, describing it, and sharing it amongst your collaborators. So as I said, all the data from the framework initiative will be uploaded there, so you can access it. It does require, there's no point storing data unless it's described in a way we can find it again. So there is a minimum metadata standard that is required to describe the samples and data files in that repository. And those are aligned with internationally agreed standards. So when it comes to the point eventually of submitting data to international repositories like EBI or NCBI, that metadata has already been recorded. And it's just showing a screen capture here. You can browse or search for project data. And this is showing the web browser interface, but it's also possible to access it programmatically via an API. Okay, so and I'm happy for any questions as I'm speaking. So please feel free to pop your hand up or pop something into the chat. Data analysis services. We, I guess, offer those for people probably who self-identify more at the novice end. So people who like to use a web browser. And Galaxy Australia is the first of these. Gareth's going to tell us quite a bit more about that, but this is my high-level summary. So you access it through the web browser. It contains many bioinformatics tools, over 1,000. You upload data into it, you run a job, and then you download data from it. You can make these complex workflows. You can share workflows with your collaborators. So amongst your team, for instance, it's got a pretty powerful compute backend. So it runs on infrastructure that includes supercomputers at NCI and Pawsey, cloud. We also have some tools that run on commercial cloud. I think something really important to tell you is that the compute is unmetered. So basically you can use as much as you like. And I think the other good thing to just mention about Galaxy as a platform is it's being used in quite a few global projects, such as the Vertebrate Genome Project, the European Reference Genome Atlas, and also locally, so other framework initiatives such as the Threatened Species Initiative, to democratize the ability to assemble and annotate genomes. So these groups have identified it's a really good way to, I guess, build workflows that are reusable and used and useful and or able to be used by many people. And that's something I think will be interesting to explore for this initiative. For command line users, Ziad is going to talk about Ables, but Ables is basically an access system we have established that provides access to computational resource, specialist expertise, so Ziad and others, and also a whole bunch of shared bioinformatics tools at NCI and Pawsey supercomputing centers. You get a pretty decent allocation, so 100 killer service units per quarter. Zia can explain what that means. I don't actually know. But the point of this is, normally when you want to get access to these shared systems, you have to go through pretty high, a high barrier or a high bar through a scheme such as NCMAS, which is the National Computational Merit Allocation Scheme. So this is a very simple way to get access to resource on there. And I think the other important thing is to note any of your collaborators in the framework can also have access. So it's been really fruitful in other framework initiatives to be able to enable a sort of collaboration space. Ziad's also going to talk about another service we offer called Bioplatforms. If you are, or if there are bioinformaticians in your framework who are using Nextflow pipelines, it's a system basically to act as a command post to run and monitor these pipelines. You can also use it if you bring your own compute. You might have institutional, you might have commercial cloud, or you may have an Ables allocation on NCI Pawsey. All of this is able to be pretty much hooked up at the back end, but Ziad will be able to tell us more about that. Okay, so I thought I'll just highlight as well, I guess, what we have under our belt for genome assembly and annotation. So for assembly, well, the first of these is Galaxy Australia. So it already includes many tools and workflows for assembly of genomes. I've just got some of the tools shown here on the left, and there's a whole bunch of published workflows. I think Garrett's probably going to tell us a little bit more about that, so I won't dwell on that. Also important to note that on NCI and Pawsey, and this is accessible via Ables, there's a whole bunch of tools for genome assembly on those systems as well. So for genome annotation, same deal. Galaxy Australia has a whole bunch of tools already on there that can be used for automated annotation of genomes, eukaryotic genomes. Same for NCI Pawsey. But I thought I'd take a little bit of time to talk a bit about a tool called FGenes H++. So this is a proprietary tool. It's a bioinformatics package for automatic prediction of genes in eukaryotes, so genome annotation. It can produce annotations that are of a similar quality to manual annotation. It's very fast compared to some other pipelines, and there's been considerable interest in this, so we've actually negotiated a national license. So it's free to use for any Australian research group. So this is available for you. You can choose to access it via the command line at Pawsey Supercomputing Centre. It runs on virtual machines there, but it's very soon going to be available on Galaxy Australia as well for web browser access. Now, I think I've heard that this framework initiative will be intending to, I guess, generate a lot of genomes, but maybe not generate so much transcriptomics data. And I think it's probably worth noting that FGenes H++ can produce high quality genome annotations without actually needing the transcriptomics data. And it does it by referring to pre-computed, they call them data matrices from a taxonomically related species. I think the good thing about F genes H is there is a very large amount of these pre-computed matrices for fungi. So there's 236 in total, and you can see the various uh, uh, groups of fungi here. So I'm not sure what species they are or how related these species are, but I think the spread is pretty good. So I'd be interested in seeing how this might work out. I also was doing a little bit of research, and I note that the Joint Genome Institute in the US, it's based at Lawrence Berkeley National Lab. They have a fungal genomics resource called Microcosm, and I noticed that they actually use 3 tools that are included in the FGenes H++ package as part of their genome annotation standard operating procedure. So I'm pretty sure it's going to work quite well for fungi. But again, we can certainly reach out to these people and explore. But I think, we have the resource, or we have the license for FGenes H++. We have all the matrices, so that's, I think, going to be fairly useful for this framework initiative. So those were tools to do the automated annotation, and I just wanted to talk a little bit about, we also host a system that helps correct those annotations. So the automated annotations are never perfect. They always need to be manually corrected. People generally might do this not for the whole genome, but maybe around a gene family of interest. But Apollo is a system, if you're not aware, you access it through a web browser and it looks like this. People call it things like the Google Docs for genome annotation, because you can effectively collaborate on a shared document in real time, and you go and you can zoom in and actually change annotations or bases and what have you. So that's what it looks like. And so if you are interested in one day correcting the automatically produced annotations, Apollo is a really great tool to do that. But it can also be used in a different way, and some groups have used it simply to share their genome sequences and annotations for others to use. And it can be set up that you can either have public access or you can share it just with a specific group. But this is, I think, a nice illustration of how one group's used it. Again, it's in the States. This is I5K, the 5,000 insect genomes. So this is their website. They have all these genomes, and I've just got 3 examples here. So when you click on those links, it opens it in Apollo. So again, this is an option for your framework initiative. So Apollo can do this, and to make it easier, we run what we call the Australian Apollo service. So basically, we take care of all the setup and the back end and the computer systems. It's resourced to support any number of genomes of any size, and all you need to do is sign up and start using it. And the guys will sort it out, and then you can just start uploading your genome and evidence files. And that just means that you and your team can focus on the annotation and not worry about setting the system up. And it's widely used by, this service is widely used by many groups around the country. Okay, so that's a sort of very high level view of the services. We do run more, including we have, I think, proteomics and metabolomics. So there's also those types of tools in Galaxy Australia, and we can talk about that maybe at the end as well. And we have other services. As far as bioinformatics training goes, we have organised a National Bioinformatics Training Cooperative, and we run webinars and workshops. So there's a lot of events. These are the past several years. I thought I'd just highlight ones that I think are, the types of things that may be of interest to this group. So these, I guess, all these workshops are run for a national audience. So we advertise it and people register. They're all oversubscribed, but these are some examples of workshops we have run. So for instance, with Galaxy, it's like, a sort of introduction to Galaxy. We've also run workshops on how to do a hybrid de novo genome assembly using Galaxy, and then also workshops around using Nextflow pipelines, for instance. But there's a large number of workshops. I guess it's worth noting that the Biocommons, we don't have the trainers. We rely on the community to do the training, and we work with this cooperative to do that. But there are options that we might be able to, for instance, think about specific training events for the framework initiative. It's also worth noting that we've worked with some framework initiatives to develop training events or modules that are both relevant to the framework project, but also a wider audience. So the first example is the Genomics for Australian Plants. And you can see here that they were doing some training workshops in related to the Australasian Systematic Botany Society conference in 2021. And so we helped them to deliver 2 webinars that were required. Participation in those were required prior to attending the actual workshop at the conference. So we've worked with that group to deliver those webinars. Another more recent example that we've helped the Threatened Species Initiative to create a bunch of videos. So they have a whole load of training modules they have. In this particular initiative, I guess the users of the data ultimately is meant to be group or individuals like conservation managers. So they're really starting at a sort of very basic level of understanding of what is genomics. So there's this a whole bunch of modules that the Threatened Species Initiative have developed, and we've helped them develop that. And we also host those on our website. There's also a whole heap of self-paced training material out there that are useful and can be used at any time. So the first of these is training modules that run on the Galaxy platform. And so these generally run on Galaxy Australia platform, and Gareth can comment on that. But you can see, and these have been developed from a global group of individuals have been developing these, and there's a lot of material. So there's 17 tutorials just around genome assembly. There's 32 on proteomics in Galaxy. There's 8 on metabolomics. So there's a whole load of those. And we also have what we call how-to guides. So these are very, I guess, specific guides on how to undertake a specific task using Galaxy Australia. So there's an example here of doing genome assembly with Hi-Fi ASM. And I just thought I'd end on also talking about, we have some community activities. So we encourage everyone to attend these. These are quarterly meetings, and they're informal, where a very wide, diverse group of Australian researchers and bioinformaticians come together to talk about computational infrastructure and services. And so there's some examples. These are coming up, one in proteomics, one in bioinformatics workflows, one in metabolomics, but we also have them around genome assembly, annotation, workflows. There's lots of those. And it can be valuable to go along to those to meet other bioinformaticians working in different projects, and also to learn about infrastructure that's available. So that's me. The take-home message is, I would say that we operate multiple robust services that will support the work of this initiative. We cater for novice and expert bioinformaticians. We will work with you to help you get up and running. So again, there's a whole bunch of service teams, and also us as well. And it's not just sort of, well, here it is, and you can, it's up to you. We will actually collaborate with your team, and we want you to get the most out of these services. I also encourage you to join our community activities, and really, that was sort of a high level view of what we already offer, but we're always interested in, well, actually, is there something else that's still required? We're always happy to hear that.